Uh, Good afternoon, everyone. We come back yet to another session. Of course, this is our section B, continuation of the uh, Bio 111 test 1 2024 that we're from doing. And this time around, these are essay questions that, of course, I'll just show you how to answer questions, and also I'll be explaining some cardinal things that you're supposed to know about this particular uh, 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 to be precise, a question that they'll ask you. So the first question is saying, why are triglycerides considered effective for long-term energy storage? But before we answer that, there are some high yield essay topics which I would want you to cover. So let me just write them. So I would say high yield. So make sure that you cover these ones for your section B or section C. So the first one which you should cover is simply the properties of water. So make sure you know this. Properties of water. So make sure you know them by heart. The second one is simply types of intermolecular forces of attraction. Types of intermolecular forces of attraction. Classify them, make sure you are able to give examples under each type. Okay, make sure you are able to do that. Then also look at carbohydrate structures or polymers such as starch. Look at cellulose and also carbs and their classification. So you need to know how to classify, and also you should be able to give at least an example over each classification that you're going to give under carbohydrates. Okay. The fourth one which I would want you to know is simply protein folding. So I'll say protein folding and phases of protein structural formation. Protein structural formation. So this includes the primary, the secondary, quaternary, the tertiary. Please make sure you know those things, okay, and be able to explain them. 5, of course, properties of proteins. Properties, or let me just say the properties of amino acids. Yes. Make sure you know at least know 5 of them and know how to explain it. Okay, zwitterion, those things, buffering system, whatever, those things, know them by heart. Okay. Just make sure you know how to list and explain a point. Properties of amino acids. You should be able to know that. Know something about lipids. Lipids, we can know triglycerides. Triglycerides. Also, you should be able to know how the bond that is found in lipids, bonding in lipids, or linkages. I would also want you to know their functions. Another important thing they will ask you is the phospholipid bilayer. Phospholipid bilayer. So make sure you know these S's and some of the things that you're going to add up. But these are high yield. By high yield, I mean they can ask you questions over this. Okay, so let's start. We start with the first question. The first question is saying, why are triglycerides Why are triglycerides considered effective for long-term energy storage? Why are they considered effective for long-term energy storage? So triglycerides are considered effective for long-term energy storage because they have a very high energy density. That is number one. They've got a very high energy density, and at the same time, they are metabolized slowly. So energy depletion is very, very low. Okay, and that makes it to have to be a very good component for high energy storage. Okay, and at the same time, they are also ideal for sustained energy needs. So I would say they are effective for long-term energy storage because because they have a high energy density and are metabolized slowly, making them ideal for sustained energy needs. Okay, that is how you answer the first question, because they have a high energy density and are metabolized slowly, making them ideal for sustained energy needs. Go to the second one. Mention two main molecules which make up a triglyceride. So triglyceride is actually made up of two. So you're going to have a glycerol, and of course you're going to have a fatty acid. So of course you're going to have glycerol. Glycerol. How do you write glycerol? So glycerol is going to be written like this. This is going to be the structure which I would want you to know. Okay, so it's going to be a carbon there attached to hydrogen, and then you are going to have an hydroxyl group there, a carbon, a hydrogen here, hydroxyl here, a carbon there. This is a triglyceride. Tri means 3 carbon atoms, so you're going to have a hydroxyl group here, and of course a hydrogen atom here. So this is called a triglyceride, and of course there are two. So there's glycerol. Sorry, this is glycerol. Then you're going to have a fatty acid. You're going to have a fatty acid. Fatty acid. So the fatty acid is going to have this particular structure. Now, we know that the fatty acids are actually long. So we usually summarize them in this way. Just write the R branch implying that it keeps on going. Then you're going to have this hydroxyl group. And of course, yeah, this is carboxylic acid group like that. So this is going to be the structure of a fatty acid. These are the two main things that make up a triglyceride. It's glycerol and fatty acids. Glycerol and fatty acids. So the two, when they react together, when they react together, and this is how they are going to react. So you are going to have a carbon here, and then a carbon here, and then you're going to have a carbon here. So remember, this is hydrogen, this is hydrogen, this is hydrogen, this is hydrogen, and then you're going to have a hydrogen here. Then you're going to have an hydroxyl group here. Hydroxyl group, hydroxyl group. Remember, it must react with a fatty acid. So when they react with a fatty acid of this particular type, okay, let me just say this is going to be a fatty acid. Let me just put one prominent fatty acid here, which I can put like that. And then I'm going to have an hydroxyl group there and the other group. What exactly is going to be formed? So this hydroxyl group, make sure that you know what is going to happen. This hydroxyl group can react. I'm going to just illustrate for this part. They can react with that hydrogen, and together they are going to form H2O. Okay, they'll form a water molecule, but then eventually they are going to form something which is going to look like this. Okay, plus something which is going to look like this is carbon, carbon, and then a carbon, and then we're going to have hydrogen there, a hydrogen there, a hydrogen there, a hydrogen there, a hydrogen. Then please don't focus on this part. This part will also react with an amino acid, a fatty it's going to react with the fatty acid, but the part which I reacted is the middle carbon atom. So that part, it's going to have something like this. This bond here is going to just attach directly to the carbon like that. Yes, and then we're going to have a carbon, and then we're going to have a double bond there. Then directly it goes on to attach to the other group. So notice what is going to be formed. You are going to form, okay, you're going to form, okay, sorry. Notice what is happening if you remain with oxygen here. So I'm going to have oxygen there attached to this guy. So at the end of it all, you're going to form a long chain. So this thing that we have formed here is called a triglyceride. So even here, you're going to have something which is going to look like this. Even here, you're going to have the same thing. It will be long like that. This thing is called a triglyceride. That's how you form a triglyceride. Okay? If they ask you to form the structure, then they say name the type of bond formed between the two molecules. So the type of bond which is formed between the two molecules is called an ester bond. Okay, it is called an ester bond. Ester bond. So this bond is called an ester bond. Why is it called an ester bond? Yes, it's because, let me show you, it's the one which has been formed here. That's how you show formation of an ester bond. Notice what you are forming. You are forming something like this here. So you're going to have this part here. So this part is simply a carbon here. This carbon is going to be attached to the oxygen here, and then it is attached to the oxygen there, and then you're going to have the other group. This is called an ester bond or ester linkage. Okay, this is an ester linkage. The bonds are here. So make sure you take note on how to form those things. Okay, so I go to question 4. Let me get a new slide there. Question 4. Question 4 is simply saying phospholipids are referred to as amphipathic molecules. What does this mean? Amphipathic molecules simply means it has both hydrophilic, so it's going to have both hydrophilic, it's going to have both hydrophilic, or hydrophilic means water loving, hydrophilic water loving properties, and at the same time, we have hydrophobic. Hydrophobic properties. Hydrophobic means water hating. So this is simply what it means when you say amphipathic. It means a molecule or a compound is going to have both hydrophilic and hydrophobic properties, both hydrophilic and hydrophobic properties. Take note of that. Okay, so we go to the next one. They're saying explain the significance of this property. Explain the significance of this property. But before I go there, I would like to explain to you something about what does it mean when you say hydrophobic, hydrophilic. What do we mean? So this is the concept here. This is where it came from. It came from what we call a phospholipid bilayer. Phospholipid bilayer is what bilayer is the concept we use when we're trying to explain how the cell membrane is formed together with its structures. Okay, so when you look at the structure, remember that cell membrane is formed by of course different fatty acids. You find glycoproteins, different proteins, carbs and stuff. There are these hydrophobic tails and of course hydrophilic heads. So these together they form what we call the phospholipid bilayer. So you are going to have something that we are going to be calling as the extracellular. We're going to have an extracellular here, an extracellular area. Of course, this area, there's water, H2O, there's water there. Then you're also going to have another area, the inside part. This area is also containing water. Then you're going to have something like this. This is how a saturated, this is how, of course, these molecules which are going to be forming the cell membrane are going to be formed like. So you're going to have something like this. This is structure. And then you're going to have something like this, going to be a saturated fatty acid. This is a saturated fatty acid. You're going to have an unsaturated fatty acid. An unsaturated meaning to have a double bond. It can be a cis or trans, like that. These guys here are fatty acids. Fatty acids, most of them are not soluble in water. They are not soluble in water. So because they are not soluble in water, they are H2O fearing or hating. This part here, I won't draw it all, but is going to contain a phosphate ion. Okay, a phosphate ion. Of course, this is a phosphate. You might have something like this. And other structures inside. Okay, you can check it up, but at the end of it all, it's going to be negatively charged inside. Remember that water has got positive partial charges, negative partial charges. Even this guy, if you go deep, you're going to discover that it might also have those charges. So what usually happens is, if this is water, the extracellular part of the cell membrane contains some fluid or water, intracellular some water also. This guy, the head, which is the hydrophilic head, this is called the hydrophilic head. This guy here is going to start getting attracted to the water molecules because these charges are going to start being attracted to the partial positive charges like that. So eventually, because these guys here are water fearing, you expect that where there is water, you expect that is where these heads are going to face. They are going to have something like this. because of that attraction. Because this part here does not contain water, these these tails are going to be attracted to this part. Okay? So this part here is called hydrophobic tail. Hydrophobic tail. Even this side, you are going to have these heads attracted to the water molecules. You can expand from this concept explaining the phospholipid bilayer. This is how the cell membrane is going to be formed. Like, where there's water, that's where these guys are going to be attracted to. The question continues. I was just trying to explain for the sake of learning. Explain the significance of this property. What is the significance of this? This that were formed here. What is its importance? The importance of what we have formed here is this. Number one, it is what enables selective permeability. Is what enables selective permeability of the cell membrane. That's what enables selective permeability of the cell membrane, allowing certain substances to get in and denying others, and also denying other substances from leaving and also enabling others. Not only this, it also enables membrane function and stability. Enables membrane function and stability. That is one thing that you must be able to understand about this particular part. We go to the next section. It's saying, which lipid type is the precursor for steroid hormones? Okay, cholesterol. What is that? That is 6. So in here you find cholesterol. They like it in MCQs also. This is cholesterol. What is the main function of lipoprotein in the body? Transportation, right? The main function of lipoproteins is transportation of lipids. Okay, let me just say transportation of lipids. Let's say particularly triglycerides and cholesterol. When you get into medicine, you understand that actually cholesterol is a very bad thing. Common causative agent for things like coronary artery diseases, atherosclerosis, and other things. Okay, another important function is regulation of lipids. Okay, regulation of lipids. What do we imply? We imply usage and storage. That's it about this particular part. Okay, so I'm giving you this one. They're saying, look at this question. They're saying classify polysaccharides. Classify polysaccharides with examples. So you classify them. Then, of course, this one is your question. You can check it out and illustrate the formation of starch, please. And cellulose, glucose, and galactose. So if you've been asked to illustrate how starch is formed, just know how glucose, of course, is formed. It's actually very simple to form the structure. Okay, just know how glucose is formed. Sorry. Just know how glucose is formed. It's actually very simple. Just put your hydrogen here, your hydroxyl here. Put your hydrogen here, your hydroxyl here. Here, inside, just put your hydroxyl, put your hydrogen outside. Here, your hydrogen inside, your hydroxyl outside. Like that. Then you react with the same thing. It's not hard. Like that. You put your hydrogen here. You're going to put your hydrogen, your hydroxyl here. Hydrogen, your hydroxyl here. You can put your hydrogen. Of course, you can put your hydroxyl here. It's okay, whichever. Then here you can put your hydrogen. This is the only part where you're supposed to exchange them. The one hydroxyl inside, hydrogen inside, or the other. Of course, when they react, you lose water. So what is going to be formed is water and the structure, which is going to look like this. Of course, you're going to have your this is just for illustration. I just want to show you that it's actually very easy for you to form these guys. So you're going to have something like this. It's going to be hydrogen. It's going to be oxygen. And then it is going to be attached to this guy. Okay, so it's going to be like this. I'm forming starch. So make sure you are able to bring out this. They may ask you, that is what you call a glycosidic bond. So that is all about formation of, let's say, starch. Cellulose, just get glucose and galactose form, and then make sure you know how to classify polysaccharides. Found this video interesting and helpful? Consider subscribing, hitting the like button, and don't forget to share. This is the mentor Abel Clemens signing out. Thank you so much.